We're going to do a short Q&A panel. So if I can invite Marnie Blewitt and Rob Nicholls, if you'd both like to turn your cameras back on again. David, I thought we might start with you. It's a question that rolls off the end of your talk nicely. So we have someone who's asked about the interplay of Prader-Willi syndrome and other missing genes in the same individual. And the example they've provided is a person with Prader-Willi syndrome who also has a small deletion in chromosome 4. I wonder if you'd like to make any comment on mutations outside the PWS region.
I think with the International Congress of Genomics, I've been talking to a few people from US about this particular aspects, and you were also part of this conversation, Diane. So the idea is it's not just the imprinted region. The human heterogeneity in the phenotype is regulated by other genes as well. And if you have other genes that will contribute to your phenotype, and obviously if you have something is deleted from another chromosome, it will contribute to the phenotype, and it will look different from Prader-Willi. It will be a new picture that has been described previously, or there might be other individuals that you don't know about. So the idea is our transcriptomics data being overlaid onto whole genome sequencing, which will tell you different hotspots that might contribute, more common hotspots that might contribute to more common issues that are observed in Prader-Willi that cannot typically be explained by what is found at the imprinted region. I don't know whether I've explained that or not, but yeah.
Yeah, it's scientifically fascinating and at the same time immensely challenging, I think, for people living with Prader-Willi syndrome. That you know, as you say, that the variability across the genome can mean multiple hits. Okay, moving on to our next question. Have we got Rob's video on as well?
Yeah, it seems to. There we go.
Excellent. All right. So a question for Rob about the AAV vectors. So are you doing any work on how to get the AAV vectors into the hypothalamus?
So right now we're concentrating on the pancreatic beta cells because we both have the cell model in which we've shown functional defects, the insulin secretion defect and the defect in a number of specific proteins, and in the mouse model in which again we've shown endocrine defects from late fetal development into the postnatal stage, and then we're looking at survival and the hormone levels. And so right now we don't have any hypothalamic phenotypes to assess, but so the AAV we use, different AAVs can infect different tissue types based on what's called serotype. So the one we're using targets specifically beta cells, but there is an AAV specific serotype that targets neurons and that crosses the blood-brain barrier. And so we do plan in sometime in the future when we're able to move on to other animal models and be able to look at neurobehavioral features of actually using AAV that would cross the blood-brain barrier. But at the moment, we don't have a specific assay to assess that. So we're concentrating on to really show proof of principle in the pancreatic endocrine phenotypes that we can actually directly address.
Having those links is really important, isn't it? Having the phenotype to test.
Exactly.
I should also invite the panelists, if you've got anything to add to these questions, feel free to flag that as well. I think there's also some really interesting work being done around the world on delivery. It's clearly a challenge. It's something that's been mentioned in a couple of talks already. Yeah. Marnie, a question for you. So it's been asked in a couple of different ways around whether gene therapies will apply to all subtypes, and someone has also asked specifically in regard to SMooCD1. So I guess both a specific and a general question, but both around who will this impact?
Yeah, so can you hear me? And hopefully you can.
Yes.
Yep, great.
Yes, we can.
So for SMooCD1, I probably am better qualified to answer rather than the general question, but yes, the intention is that it would be for all subtypes. So whether it's UPD or deletion, or whether it's an epigenetic, so an imprinting defect itself, we expect that it would be equivalent and appropriate in all cases. This is something obviously that has to be trialed, but for the moment, the data that we have from the lab suggests that it would be appropriate in all subtypes. So that's exciting. I think for other types of therapies, then again, I think most people are aiming to treat all patients rather than one particular subset of patients. But again, as I said, I think it's always really important that that they need to be trialed in, at least in the UPD versus the deletion patients, so that you know that that's actually a fair thing to do and it's going to be equivalent, because there's a difference between us thinking that will be so and then the unexpected consequences. Yeah. So it will be that once we get to clinical trials for any types of therapies, it will be important that we have patients of both types, and ideally also the imprinting defects as well. But of course, that's a much rarer population.
Mm-hmm. Yeah, fantastic. Rob, do you want to add anything further to that?
No, she's covered it extremely well. So
excellent. It's interesting. I think, Marnie, that you're raising that stratification again, which David referred to as well. That idea that that Prader-Willi syndrome has that complexity of subtypes, but potentially also, the role for other biomarkers, which David, you might like to comment on in terms of that role in clinical trials as well.
Right now, you want me to comment on it? Okay.
If you'd like to, you don't have to.
Yeah, sure. So I'd be very interested to look at clinical trials that have already gone through in Prader-Willi. They may not have been successful based on analysis of Prader-Willi as a whole, and have a look at their genomics and see what are subgroups that have responded. Because even if, it would be a waste if biology and animal studies make sense, and it's not successful because humans are a lot more heterogeneous than mice. And so being able to identify subgroups of individuals where it might be beneficial, it would be better not to have a treatment at all. So I think it'll be a very interesting project.
Yeah, and to add to that, I would say that it obviously it goes beyond even, I was very broadly classifying into the three groups that we normally think of, but it's way more than that. And even with the deletion patients, where are the break points? And this could really influence how patients respond. So as David says, you need as much nuance, I think, as you can get, ideally. And of course, that makes much larger clinical trials, which is also challenging, but it might mean you can find the right thing to do. Yeah.
To be able to answer these questions, you need to have more participants to understand the natural history, right? If you got 2 participants, you won't be able to stratify things very well. So the more participants there are, and the more questions can be asked in terms of these subgroups and the mechanisms that are specific to them, not just the subtypes.
Yeah, absolutely. A very broad question. So maybe just pop your hand up if you want to take a stab at it. Is stem cell replacement for gene 15, so chromosome 15, I presume, a possibility? Stem cell replacement. I presume they're referring to some of the recent work around sort of regrowing the hypothalamus or sort of regenerating the whole of the hypothalamic organ rather than an individual gene or reactivation. I don't know, does anyone have a comment in that space?
I can make a comment, but I'm not really well qualified. I feel like I can always talk, but it's my personality. But I would say you probably need a stem cell biologist to answer that really clearly. My one comment would be that it probably may in the future be possible, but we'd need to be well in advance of where we are now, because the methods often to create those kind of organs or organoids sometimes often leave behind cells that are of an embryonic origin that, if they're put into a patient, would end up creating a brain tumor. And so, you don't want to solve one problem only to create another one that's going to result in rather short-term lethality. Of course, that would be terrible. So I think there are some things that they're working on, but in general, this is true for all of when you think about stem cell therapy in general, these are issues that need to be solved. It's not specific at all to the hypothalamus or specific to neurodevelopmental disorders, they're big problems for the research community to solve.
And I think I could add to Marnie, and the thing with stem cell therapy when you're talking about the brain and hypothalamus is it's very invasive. And so stem cell therapies are much more feasible, I think, for tissues in the extremity that are not going to be as invasive. Whereas some of the other techniques, if we have small molecules or antisense oligos that can activate broad cell types that might be affected in Prader-Willi without that invasive nature, I think that's more likely to be the future.
It's probably safer in the long run. Yeah.
It's certainly something we've put on more of a watching brief list at the moment. Let's see how the technology unfolds. Keep it on our radar, but as you say, the safety questions are very real. Biobank question for you, David. Will the DNA submissions need to be ongoing, or will it just be once and done, including behavioural assessments and intelligence assessments? Will these tests be repeated as patients age, or is that irrelevant?
Great question. Well asked. So right now we have ethics to do it once, and so in the future, if that happens, I guess I cannot rule it out. But right now we're just interested in doing it once. And if we do intellectual functioning assessments and we provide the reports back, the families might want to use those for other purposes. Or, for instance, if you already had ADOS or IQ assessment done for school or some other purpose, we won't do assessments again. We'll just use what has been already done. So the time that is spent would be a lot less, and many of the families would have had it done already. So I hope I answered that question.
Yeah, it sounds good. So I think if I could paraphrase as well, so it's always great to have longitudinal data, but it just getting that single first point done to start with is a good thing. Right. And it's, I hope I'm not overstretching, David, but I believe you're looking to recruit a few people up in the Brisbane area in the next little while. So I believe Emma's got 1 or 2 spots. So if we have families in Queensland, feel free to reach out. Do you want to add? Is that okay, David? Is 1 or 2 all right?
Yes. Thank you for your help with Queensland, and thanks to Hani and Michael for their assistance. Much appreciated. But yeah, Queensland is now or in the next week, and we're finalizing agreements with New South Wales. Hopefully, we'll be done in the next 2 weeks, and we'll be opening New South Wales, and then we are gonna have things happening in Victoria as well. But don't. If you're interested, email us. We'll archive it, and as soon as it opens up in your state, we'll get in touch.
Yep,
so don't
fantastic.
wait until we send a message saying, okay, go in New South Wales or Victoria. You can send it to us now. We'll archive it and get back to you in a month or 2, or maybe less.
Yep. Excellent. And we'll be promoting some other clinical trials in the latter part of the session with some resources for finding contact information for David and his team there as well. I think, so two last questions. So for Rob, why do some countries have higher levels of UPD?
Sorry, I missed that.
So at the start of your talk, you mentioned that some countries have a higher incidence of UPD. Would you like to comment on why that might be?
Yeah, so in I think there's been reported studies from Switzerland. Again, we're in countries or societies where people are having children at a later age to establish career and so on. Then with the association of advanced maternal age with the uniparental disomy mechanism, then in those countries, the frequency of uniparental disomy is a much higher incidence. So I think in Switzerland, they published up to 50% of cases with Prader-Willi were now uniparental disomy. And so it's associated with the advanced parental and in particular maternal age.
Yeah, definitely a challenge. Okay, so to finish, I've got one question I would like to pass to each of you in turn. It's the obligatory "how long is a piece of string" question. So we've been asked, what is the time frame for your research to reach commercial release? So as I say, how long is a piece of string? Rob, would you like to comment on your research?
Well, in terms of our AAV gene therapy approach, which really is largely at this stage, we're not trying to attempt to get it to clinical trials. We're using it more as an experimental approach in cell models and in animal models to really be able to dissect which genes are important for which phenotypes, and can we, by proof of principle, show that by replacing genes, we can correct those clinical phenotypes we see. And because if we can take a 8-gene AAV or a subset of genes, we can then determine exactly which genes contribute to which phenotypes. In terms of commercialization and any clinical application, that's well into the future. It's going to take a couple of years to get through the cell and animal models. And in terms of approach, the AAV, we can fit 8 of 10 in, but we can't, for example, fit MAGEL2 in. That's a large gene that needs its own AAV. And so approaches that use small molecules or ASOs that can reactivate the maternal allele may get to the clinical trial and commercialization sooner, or be more applicable. But we have to see, again, as others have mentioned, we need multiple approaches, and this is an alternative approach that certainly has a high significance from understanding the genes and the functions. Does that answer? I hope the question.
It's a tough question to finish with. So I think you've led into Marnie's work very nicely there. Marnie, how long's your piece of string?
Yeah, it's really hard to commit to how long it is, especially now because we have these 2 portfolios, if you like. So and they have different time courses, I guess. I think if things went really well, it would be between 5 and 10 years, but this is a guess because in my experience to date, drug discovery doesn't go really well. Drug discovery goes incredibly slowly, and what you do is you find the next hurdle you need to overcome. So I think that's if everything went well. But I'm ever the optimist, so I think that it is possible, and I think that you keep trying to find the best way to develop the best drug. And we're at the moment both in validating SMooCD1 as being a relevant target, as well as in drug discovery. So we're trying to do it as fast as possible by progressing both fronts. But time will tell, I guess.
It will, and I guess highlighting what you raised as well in terms of the importance of understanding those mechanisms in terms of bringing in the company support as well.
Yeah.
So yeah. So David, you've got a slightly different data set that you're working with and slightly different opportunities. Would you like to comment on timeframes to commercial release for your research strands?
We were finding a lot of new things that haven't been previously described, but we're not planning to patent protect any of it. Our strategy with this work is to make it available to the research community as quickly as possible, because there's so many questions that can be answered with this rich data set. And there's only a finite number of brain tissues that are available to answer the questions, so many questions. So the idea is we hope to publish end of this year, and we hope to publish end of this year. And as soon as we have done that, we will make things available to the general research community. The bit that I have not presented on is drug repurposing. And that would a lot quicker than having new drugs. So if there are drugs that are being used, for instance, we do have some information about the immune system being affected in the difference between deletion versus non-deletion. So if there are specific component pharmaceuticals that are being used to address a pathway that is different between deletion versus non-deletion, that is already in practice, it's a lot easier to make mechanistic argument into preclinical and clinical trial than having something being worked out from scratch. So, and there are a lot of leads, and the question is prioritizing where we go, and we won't be able to do all of the work. It will need to be made open to the broader research community. So that's our strategy.
Yeah, there's a massive amount of potential from the data that's being generated, most definitely, and the repurposing pathways will be interesting too. which I think what not only have I enjoyed the science of the talks, but I've also enjoyed the diversity of the approaches that you're all taking. And I think in that lies the potential to get therapies and have impact as well.
I think later today you'll have Teresa from FPWR. I think you should ask her this question about drug repurposing and what they're doing with this.
We did indeed, David. We did indeed. Excellent. All right. So I just want to again offer my thanks to the three of you, Rob, Marnie, and David, for sharing your research with us today. It's been wonderful to hear the updates and see where your research is at.